फटाफट जल्दी आगे आ जाइए, ये सब लास्ट में है। आगे आ जाओ आगे। इतनी बार घुटने के लिए चाहिए। अलग से इन्विटेशन देना पड़ेगा। पूरा कवर करो। आगे आ जाओ आगे। हाँ पूरा कवर करो। आगे आ जाओ।
I, Samuel Moga, working as assistant professor in the Department of Pharmacology at ISS College of Pharmacy and Autonomous College, Moga, Punjab. It's my great pleasure to welcome you all in one-day national workshop and hands-on training on 3D bioprinting technology and therapeutic applications, sponsored by Selling, a bio company in Sweden, and ATCG India, and organized by ISF College of Pharmacy and Autonomous College Moga, Punjab, under the aegis of Internal Quality Assurance Cell. So for this, we have two experts, Dr. Santosh Kumar, Application Scientist, ATCG India, and Ms. Shivani Tyagi, Product Specialist, ATCG India. The chief guest of the day, Secretary ISFCP and Chairman TLF, Engineer Jinesh Gargji. I, on behalf of ISFCP family, welcome you, sir and ma'am. I would like to extend my welcome to Director Sir, Professor Dr. G.D. Gupta, Vice Principal Sir, Professor Dr. R.K. Nanak, all HODs, faculty members and dear students. Kindly allow me to express my gratitude toward each and every one of you present here for sparing your valuable time. So let's begin the auspicious day with traditional lamp lighting ceremony as a tribute to Ma Saraswati, the goddess of knowledge. So I request our guest, Dr. Santosh Kumar, Ms. Shivani Tyagi, Secretary ISFCP and Chairman TLF, Engineer Jinesh Gargji, our Director Sir, Professor Dr. G.D. Gupta, Vice Principal Sir, Professor Dr. R.K. Nanan, and all the HODs to come on the stage for lamp lighting ceremony. Now I request our Director Sir, Professor Dr. G.D. Gupta and Engineer Janesh Garji to please come forward for the floral welcome of our guest Dr. Santosh Kumar, Application Scientist, ATCG India.
वायरलेस वाला है, वायरलेस वाला नहीं है।
Thank you, sir. Now I request our Vice Principal, Sir, Professor Dr. R.K. Naran and Professor Dr. Shamsher Singh, HOD, Pharmacology, to please come forward for the floral welcome of our guest, Ms. Shivani Tyagi, Product Specialist, ATCG India. Thank you so much, sir. Thank you, ma'am. Now I request our Director, Sir, Professor Dr. G.D. Gupta and Vice Principal, Sir, Professor Dr. R.K. Naran to please come forward for the floral welcome of Secretary ISFCP and Chairman TLF, Engineer Janesh Garji. Thank you, sir. Now I invite our dynamic and most energetic Director, Sir, Professor Dr. G.D. Gupta on the stage for the welcome speech.
Good morning, everyone. Good morning, sir. Good morning, everyone. Good morning, sir. Sarve bhavantu sukhna, sarve santu niramaya, sarve bhadrani pashyantu, ma kasya dukh bhave. Honorable, respected, today's chief guest, very dynamic, energetic, and dedicated for the profession, Engineer Janesh Garji, Chairman ISF Educational, Secretary ISF Educational Society, and Chairman TLF, a global school. Two guests and experts among us, Dr. Santosh Kumar ji and Ms. Shivani Tyagi ji, Professor Dr. R.K. Naran ji, HODs, organizing committee members, all faculty members, and dear students. First of all, from core of my heart, I welcome today's chief guests, experts, all HODs and faculty members in this conference room. The purpose of this conference to give a brief about the 3D printing and the application of 3D printing. There is a number of questions. What is the application of 3D printing in pharmaceutics, pharmaceutical chemistry, pharmacognosy, pharmacology, because all STEAM students over here. I want to give some of the examples. In those days, 3D printing technology in the treatment of the cancer as well as the so many diseases. I have attended first workshop in NITR, Chandigarh. At that time, they have demonstrated some of the 3D printing technology concepts. I met one of the person, he is the taken one of the CT scan and so many other diagnosis things. After that, the final is need to the operation. So nowadays, exact position, location and how it can be operated. So doctors take the help of the 3D printing. As you know, here is the SUTEX QA regulatory students over here. So if you want to know what is the hardness of the tablet, so hardness of the tablet at which place you have to calculate. Center place, side place, between and inside like that. So at that time we are using the lead ball for the determination of the hardness at the different points. Nowadays, with the help of this one, we can find out what is the actual position. I want to give over one of the example. मैं अपने बेटे का देना चाहता हूँ. उसको पाइल्स की प्रॉब्लम हुई, डॉक्टर को दिखाया, डॉक्टर ने कहा नेक्स्ट डे आई विल ऑपरेट, और तब उन्होंने 3D ड्राइंग करके उन्होंने चंडीगढ़ से मंगवाई, 3D प्रिंटिंग मंगाई, और उसमें एक एक चीज दे हैव टू द मेंशन क्लियर. So gentlemen, nowadays most of the conferences and everywhere, this is not essential that you are in which branch you are working. But this is important how you can utilize this technique. Initially, this technique was applicable in the electronic, electrical, and highly applicable in the robotics. Nowadays, this is in the form of the diagnosis of the disease, treatment of the disease, in the operation, and highly applicable in the cancer-like of the disease during the operation, diagnosis, as well as the identification. So you can listen very carefully. All lectures, so this is not as simple as the machine. The cost of the machine, our expert they will discuss how it can work, and they demonstrate some of the experiment with the small groups. So this is a big opportunity. Dr. Siddharth, I am taking the responsibility and invite the company in this institute for your purpose. So this is the whole day activity. I am very surprised to say that our 3 faculty members, they have given the full input to shift from the back seat to the front seat. बड़े दुख की बात है. मुझे बड़ा दुख होता है. आप पीछे सोना चाहते हो, आगे जागना चाहते हो. आप आगे भी सोना चाहते हो तो सो सकते हो, सोते रहिए, क्योंकि किसी भी चीज को उसको सुनाया जा सकता है, सोते हुए को जगाया जा सकता है, जागे हुए को सुनाया नहीं जा सकता अगर वो सोना चाहता है. आज whole of the system is the doing the work for the students and you are away. मेरे टाइम में तरसते थे ये कॉन्फ्रेंस अटेंड करने के लिए, शिद्दत लगती थी, because at that time there is the no availability or very less availability of the infrastructure. पूरी कंट्री में 2 ही कॉन्फ्रेंस होती थी. One is the IPC, another one is the APTI. APTI for teacher and IPC for the industry. And you can take the participation. Nowadays, many expertization over here. So I want to include one thing over here is the also, if you can go through the infrastructure or like the system of the some of the university, how they are doing the work and what is the need. कल मैं एक कॉन्फ्रेंस में था, मुझे बड़े दुख के साथ इससे कह रहा हूँ, आज भी मैं बड़ा बेचैन या मन से कह रहा हूँ कि हमारे कॉलेज का पूरा झंडा गड़ा हुआ, पूरा झंडा के एक भी मिनट ऐसा नहीं था या 10 मिनट नहीं था कि आईएसएफ नहीं आ रहा हो. लेकिन जब एक चीज आई कि पूरे कंट्री में सिर्फ 2 ही ऐसे इंस्टीट्यूट निकले जिन्होंने मोर देन 40 स्टूडेंट जीपैट में एक शॉट में निकाले. आई सरप्राइज एंड आई एम थिंकिंग अगेन एंड अगेन. So gentlemen, this is the right time. Our faculty member, our management, we are dedicated for the students. अगर कल नहीं हुआ है तो आज देर मत करो, आज करिएगा. अगर आज देर कर दी तो फिर कभी नहीं हो पाएगा. Those students unable to clear the GPAT in the B-Farm doesn't matter. मैंने भी नहीं किया था, लेकिन ये तय कर लिया था GPAT ऐसी बड़ी चीज नहीं है कि मैं ना कर सकूं. So what is the scholarship? और एक गजेंद्र सिंह जी ने स्टेज पर खड़े होकर कहा था, गजेंद्र सिंह के इंस्टीट्यूट से 38 स्टूडेंट निकले. एक कानपुर का कोई इंस्टीट्यूट है जिससे 40+ इंस्टीट्यूट निकले. मैं सोच रहा था मेरा नाम क्यों नहीं है, और सिर्फ वो नाम इसलिए नहीं है क्योंकि आप नहीं चाहते हैं तो आज से कोशिश करिए कि अगला जो सेशन आ रहा है मैं वो करना चाहता हूं और करके दिखाना चाहता हूं, बिकॉज फुल टाइम वी आर डेडिकेटेड फॉर दिस। पर क्या चाहिए वो बताइए। सिर्फ एक चीज चाहिए कि हम करना चाहते हैं। एक और एग्जाम्पल बहुत सुंदर सा मैंने भी दिया था, दूसरों ने भी दिया था। There is no need to the more and more books and the coaching center. Not required everything. Only the required your how much time you are devoting the study and how much time you are devoting for the consistency like that. वो consistency के लिए किसी डॉक्टर या किसी की जरूरत नहीं। तो आज आप शिद्दत से इसको सुने। You can listen each and every parameters. अपने क्वेश्चन पूछ सकते हैं। This is not essential that कि in this branch you are studying। कई बार ये हमारे दिमाग में रह जाता है। हाँ, मैं जो एक दूसरा एग्जाम्पल दे रहा था, जो कई ऐसी यूनिवर्सिटी हैं, they are doing 2 project in M.Pharm. One project is the minor project and another project is the major project. तो यहाँ शमशेर जी बैठे हैं, हमारे यहाँ मुंबई से 2 लड़कियां आई। They are the student of M.Pharm Pharmaceutics और वो प्रोजेक्ट किसमें करने आई थी? कोलॉजी में करने हैं। जब हमारे यहाँ प्रोटोकॉल होते हैं कि हम शुटिंग से तो हमें चूहे के बारे में नहीं पता है, कोलॉजी को 1 साल में सारा पता हो गया। तो जेंटलमैन नाउ अ चांस फॉर द मल्टी डिसिप्लिनरी वर्कआउट, तो उस पर होमवर्क करिए। बिकॉज इफ दिस इज रिलेटेड टू यू और नॉट रिलेटेड टू यू, व्हाट कनेक्ट टू यू, अगर आप डोजेस फॉर्म बनाएंगे जब डिजीज के बारे में जानेंगे, आप एनालिसिस करेंगे जब उसके प्रोटोकॉल के बारे में जानेंगे, आप उसके ऊपर एक्सपर्टाइज करेंगे जब डिजिटलाइजेशन और लाइक 3D प्रिंटिंग के बारे में जानेंगे। सो फॉर दिस पर्पस नीड लॉट ऑफ वर्क। और मैं चाहता हूं कि आने वाले सालों में मैं भी मेजर एंड माइनर प्रोजेक्ट पर होमवर्क कर सकूं, सो दैट आज की डेट में हम सभी रेस में खड़े हुए। Everybody want a good scale. Everybody want a lot of money. Everybody want a one of the scope in the position like that. कहाँ से आएगी? There is a lot of positions available over there. What capacity you have? So try to do the hard work. And कोई ना कोई आपका इंतजार कर रहा है। Work on the quality. So again I welcome all of you. मेरी मन की बात थी, जो मेरे दिल की बात थी, वो कह देना जरूरी था, क्योंकि यहाँ पर ऐसे 80 विद्यार्थी बैठे हैं जो इस बार जीपैट में बैठना चाहिए। और मैं दावा करता हूं, अगर आप सुनिश्चित कर लीजिए, मैं 80 के 80 को आर पार कर दूंगा, ये मेरी गारंटी। आई विल इटसेल्फ विद द टाइम ऑफ द evening, morning, at time, what time you want to over there. I will be there. I will separating library का section develop करके you can give only the 3 hours devotedly only and only for the GPATs. क्या फायदा होगा, क्या नुकसान, इसकी गणना हम लोग बाद में कर लेंगे। You can enjoy of whole day function and interact with the expert as and when required. You can ask the question and during the after the second hours we can organize one of the workshop in the 3 parts में। पहले 4 पार्ट में थी, अब 3 पार्ट्स में कर देंगे। लगभग 50-60 का स्लॉट लेकर के उसको आप इत्मीनान से सुने, अंडरस्टैंड करें, क्वेश्चन पूछें, नए व्यूज दें। हो सकता है आपका एक व्यू इनकी मशीन के लिए, इनकी टेक्नोलॉजी के लिए एक नई चीज को जन्म दे दे और ये उस पर भी काम कर सकते हैं। सो थैंक यू वेरी मच एंड वेलकम टू ऑल।
Thank you so much, sir, for enlightening us with your motivating words. Now, I invite Professor Dr. Siddharth Menon, in charge IQAC, on the dais to brief out the workshop.
Thank you, Sanya ma'am. Today's chief guest, respected Engineer Jayesh Garg, Secretary ISSCP and Chairman PLF Moga, worthy Dynamic Director Professor Dr. G. Gupta, Vice Principal Dr. R. K. Narayan, all HODs, dear faculty members and students. Today, ISS College of Pharmacy is offering an interactive offline workshop on 3D bioprinting technology and its therapeutic applications, sponsored by CellLink, a biotech company in Sweden, in collaboration with ATCG India under the aegis of IUAC ISFCP. The aim of this one-day national workshop and hands-on training is to introduce the 3D bioprinting technology and its versatile application with hands-on to Cell BioX 3D bioprinter. And the basic purpose of this workshop is to generate new ideas in the field of regenerative medicines where bioprinting with mini livers or full-scale skin models are there. They are also used in cancer research, which is easy to construct tumoroids in high throughput manner for comprehensive work on cancer biology. Especially in case of drug discovery, effortlessly create robust 3D cell and co-culture models to be treated with drugs for downstream analysis and also for food production. The 3D bioprinting is also employed in bioprinting tissues and organoids, surgery preparation assisted by the use of 3D printing models, 3D printing of surgical instruments, and custom-made prosthetics using 3D printing. So this is our humble request to all students and faculty members. Please put your questions, generate your ideas, and make this workshop in a very scientific manner. Thank you so much.
Thank you so much, Dr. Menon. So, without taking so much of time, I request Dr. Santosh Kumar, Application Scientist ATCG India, to please come on this stage for the technical session.
A very good morning to all of you. Myself, Dr. Santosh Kumar, Field Application Scientist, ATCG India. And I would like to thank you, Siddharth sir, and all the faculty members and all the executives from ISF to provide this opportunity to come us and show our technology and details. We will, first half, I will try to give some overview of the am I audible? My voice is audible?
ये लाइट को करवा दो ये वाली बंद
Now it's audible? Yes, sir. Okay. So today we are here for discuss about 3D bioprinting technology and its therapeutic applications. So we are channel partner of Sterling company, which is pioneer in 3D bioprinting field, which provides all necessary hardware, software, and biomaterials for 3D printing related things. We have a range of instruments for 3D printing. So I will try to cover in my presentation, and whenever you feel some question, without hesitation, you can ask, or at the last of talk, we can discuss one by one. So it depends on you. You can ask any time. So some brief introduction about the technology. So 3D bioprinting. I think I will start with some very basic things like printing. Everyone knows what is printing. Then you add 3D printing. Then you add bio. I mean 3D bioprinting is not new thing. As sir has told that it is being used in material science from very beginning, from last several decades. Now people are using 3D bioprinting in every field of life sciences. While there are certain challenges life sciences industry facing. For example, since I am in the pharmacy college, so I will start with the pharma example. For example, studies have clearly shown that out of 10 drugs, 9 are getting failed in the clinical trial. So you can imagine the wastage of money and time. That at the end of the day, you are coming to know that this drug has not worked, or it is highly possible that that drug will work fine in animal models, but the moment it goes to humans, it will not work exactly, because we have taken the cells from the animals. The other thing, there are several lives are getting lost due to lack of organs. There are several heart patients, liver patients, lung patients are waiting, but there is no enough heart available, liver available that you can transplant it. They are waiting in labs across the globe. The other thing, if you have, if once you have bioprinted structure, you can reduce your time in checking your drug. Without final going to animal system, you can check your drug efficacy before that. And on this 3D printed model, we will try to print some structure in the second half once we make the presentation clear. So there are, I will show in the next slide, there are several applications. Yes, you can focus on this slide. Like if you see just things, so I have tried to cover some important field where 3D printing has been used for the things. For example, pointer is not good. I want to go for this. Mr. Scott, we go to first slide. Yes. Hello. Yes. So sorry. Actually, if you see this slide, I have tried to discuss about the drug testing. If you check your drug before going to final clinical trial with 3D printed model, so you will save money and time. You can check with personalized medicine field. Like personalized medicine, one company called Carcinotech, they have used patient especially they have taken pancreatic cancer cell line. They check for chemotherapeutic dosage and they have tried to understand how much dosage is needed. So you can see it is helping in personalized way that some patient might need more dose, some patient need less dose because the patient will die if they will get overdose. Sorry. So you can see the like in tissue engineering, people are making several like for example, if your heart valve is not working or some muscles is getting defected, you can generate that part and you can make 3D printed things and use that part in 3D printing print. Apart from that, it has been used in cosmetic testing. For example, after checking the skin sensitivity, you can just like company like Lakme, they have developed their products. They are checking drug or cosmetics before that while launching to the market. Like even food alternative. Nowadays in Western countries there are some studies coming out where in place of biological meat they are using the component of that part and developing biological meat. So you see the utility of this 3D printing technology, each and everything, even for space biology. People are printing 3D models, they are taking into space, they are checking the gravity effect, so how the cell will behave it will go to space. So I mean to say, this is mentioned to few, there are not many examples, there are more than 100 applications where 3D bioprinting has been used. So you can see the utility of this technique. So we have 3D bioprinting solutions. I will try to for 3D printing, Cell-Link has two types of solutions. One is pneumatic based, which is pressure based, and another is light based. So we have this extrusion based panel In the left panel, you can see the upper two models are incredible and incredible plus, which is very old model. You can see the BioX. I will try to cover in the later slide. This is BioX 6. Here, three different exchangeable print head in BioX, and in BioX 6 there are 6 exchangeable print heads. So you can use 6 different type of biomaterials in the one go. For example, if you have to print a skin-like structure, you can fill different things like for epidermis, dermis, hypodermis, keratocytes, hair follicles. You can fill all materials relevant to that, and you can print the structure. This is light-based. The light-based also resolution is more in the light-based printer, but here throughput is very less. This is for that high throughput we have Moga X, which prints in plates, 96 well plates. You can print it. On top of it, we have all necessary hardware, software, consumables from Cell Link company. I mean, you need not to optimize the biomaterial. You think of any biomaterial, we have other company, advanced biometrics from Cell Link. You just select your biomaterial, you see the composition, and you start working. We have software DNA Studio 4 for making the models. We have all necessary plastic ware, consumables, and of course this hardware, this machine. So we have all solutions from Cell Link, starting from hardware, software, consumables. You can use it from Cell Link, and you can make the things. So what is 3D printing and what is 3D bioprinting? 3D printing in simple way, you just see the making. Just 3D printing is additive manufacturing, is making layers. 3D structure, you can see, everyone heard about 3D, just making layers. You know about the 3D printing. The moment we add biomaterials, it is 3D bioprinting. That means we are printing a structure in 3 dimensions, but along with we are adding biomaterial, so that this science is 3D bioprinting. There are 2 ways. Either while printing, you can add material, biomaterial, or once you printed, you can add your cells. Like you see, there are two possibilities 3D printing. You either you add cells post printing, or you can add cell laden biomaterial and you print the live printing. So both processes are possible in this machine. We can go with either after printing you sit with cell, or while printing you can take the cells and you just print the structure. So why 3D printing is needed? Normally, usually process people are using 2D culture. It's from AJ. People are making 2D culture, but here cells are adherent. So it is highly possible that cell will not get proper oxygen or proper nutrient in the in vivo environment. So cell will be undernourished or maybe hypoxic. So you cannot correlate directly with your in vivo physical environment. So if we print 3D structure, then cell will be more close to your natural system. So in that way, it will help. See, there are some examples to show you, like just to say, this you can see this structure. If you print 3D things, liver, lungs model, the cell will be, the printed structure will be more similar to your physiological environment, and whatever any biological processes down the line, if you analyze any gene expression study or any metabolic activity or any drug testing, cell will behave more closely to your exact physiological environment. So there are two ways you can print the things. Either you can print cell as provide or make cell like a structure. One can be encouraged dependent or one can be encouraged independent. So both possibilities are there. In some cancer research, people are making as provide and they are checking their drugs. So you have both options. You can print both ways. I mean, either you can print as provide or encourage dependent, or you can print and you can check it. there are in the preclinical testing there are chances of false positive and negative. How? If we, for example, if we check with the 2D and 3D, the cell will not get, as I mentioned earlier, the cell will not cell will not feel exact physiological environment. Cell might be hypoxic to the cells. And another like in this thing, if we also mice, rats are similar to humans. I want to highlight this point. If you check your drugs in animals, it is highly possible that they will not behave exactly how they are behaving inside. On top of it, I also want to highlight the thing. There are some countries where they are making ban of animal systems to study things. You need to go for several approval. But if you have printed your bioprinted models, you can do your analysis, drug testing, all necessary experiment easily. So these are the advantages where you can use 3D, you can exploit 3D printing technology and do your analysis. This is very classical example. There are reports where they have done drug screening for colorectal cancer. In colorectal cancer, they have used monolayer culture and 3D bioprinted things. They have checked the live cell with the 2D printed and the 3D printed. You see in the right panel, the drug response and the percent live cell are more in the 3D printed things, and in 2D printed things, the percent live cells were less. Clearly, their studies have shown that 3D printed model versus 2D printed model differ in cell morphology, gene expression pattern, and drug response. They have checked the drug and they have demonstrated that 3 out of 2 drugs behaved more efficiently in 3D printed models. So you see the utility of 3D printing, and the hypoxia related genes and cell-cell adhesion related genes were upregulated in 2D printed structures, 2D printed culture cells. But if in 3D printed, they have seen they are more close to animal cell and their expression were more close to in vivo environment. So you see the utility of this 3D printed and 2D printed things. In between, if you have any question, please ask me. Don't hesitate. You can ask any questions. So, this slide is again this slide provides one complete layout how 3D bioprinting workflow works. It's not available. That's what I asked. Oh, sorry. My voice is because it's going on back side or not. Please raise your hands that my voice or something is not clear. We will solve and then we will proceed further. This slide typically shows the 3D printing workflow. There are, I mean to say, first if you have to print the structure, you need to design the model. You need to design the model, you need to select the biomaterial, you need to select the machine, you need to make it pure, then characterization and downstream applications. So, first of all, any type of a structure you can think of, you can design, or Sterling provides one repository which is called 3D Bioworks. I will show in the coming slide, where already printed structures models are available. You need to simply take it and proceed with that structure, or you can do manipulation. There, in the next slide, I will show how to design 3D models. This machine takes in 2 formats. Either you have to provide G code file or STL file. For that, you need not to be good programmer to designing the structure. We have DNA Studio 4, where you just simply think of a structure, or you can just select the structure or any shape, any dimension, and you make things happen. So in the next slide. Here I want to show some video, if it is possible. This one, yes. You will see if you want to draw the structure, you will see select the shape, design the shape. That model printed, and another I will see this video I want to play. This video will show you how this machine, the printhead works, the 3D printing, how it works. So I want to see, you see this how it's printing. Again, we have spelling. Yes, this one. This we have biomaterial, and see here in this slide I want to highlight 2 points. If you print your any structure, if you don't cure it, the structure might not be stable. You have to, once you print the structure, for example, arginate based structure you are printing, so you have to covalently cross-link it with the calcium chloride. If you don't cross-link this, in this video you might have seen that the structure was not stable. Without cross-linking, how it's behaving. The moment you add calcium chloride, you see after that with the spatula, you see the structure. After that, it is easily pickable. So after it is very important process. Once you print the structure, yes. So I will try to show some videos in between so that you can get one overview how things are working. So we will cover every step in which detail. I will try to explain how to design 3D model, because these are the basic steps. If you understand this, then you can design any complex step, any structure you can think of, and you can design it. So designing 3D model, selecting biomaterials, and of course 3D printing, and after that you need to cure it and incubation and downstream application. Here also I wanted to highlight, once you printed the structure, after that you can do all analysis what you were doing with 2D culture. So this is once your structure is printed, after it is it's almost similar to 2D things. After that you can do all downstream applications. So designing 3D model. This is very important slide. Please ask me if you don't understand anything. If you have to select things, so you just select the shape, you define the dimension. Here you have option. I think you missed, probably you can go back. I think you missed some information. I think that is covered. Just please go back once. One more slide. I will change. Yes, please. I want to explain something here. For this machine, we need, this machine takes either STL format or G-code format, right? So here STL is described the surface geometry. This in G-code files, there is, you see there are so many lines. Okay. So for example, if someone is interested in making some cylindrical structure or maybe some half cylindrical structure, so he need not to Sorry, I'm sorry. Yes. So you just simply need to change the line. You can change any one of or two, three lines depending upon, for example, you want to change the height. Simply you need to change that line. This machine will take that command and will print accordingly. If you want to change the diameter, you need to change that line and machine will take that command and it will print the things. We have one software that comes with machine called DNA Studio 4 where already several predefined parameters are there, or you can select or design material of your choice. So STL file, please play this video. You see it's taking shape. Select the shape. There are so many templates. Any depending upon your requirement, you select the shape. You define the dimension. For example, if you want to create tunnel-like structure inside the structure, it is easily doable. Simply, you need to select the dimension and you just give that command along with that program. It will create tunnel-like structure. You can define the diameter, radius, length. You can define that. You see how simple. It's easily doable. You need not to be good programmer. It's very user friendly. It comes with machine. It's free of cost. And latest they are upgrading also and making the things easily available. See, once the side has been decided, you see inside the channel like structure also they are making the program. Yes, but yes. Shivani, will you explain a little bit more? This is very important slide है.
सिंपल लैंग्वेज में कहें तो ये मशीन है और ये मशीन मतलब हमारी लैंग्वेज में समझती है. उसके लिए हमें क्या करना होता है, ये जी कोड फाइल और एसटीएल फाइल की लैंग्वेज समझती है. तो हम लोग सोचेंगे कि हम बायोलॉजी स्टूडेंट्स हैं तो वी डोंट नो कि कैसे कोडिंग करनी है, क्या करना है. तो इसके लिए क्या हमारे पास डीएनए स्टूडियो सॉफ्टवेयर है. उसमें हम जस्ट जैसे एमआरआई स्कैन होता है, सीटी स्कैन होता है या फिर कोई इमेज होती है, जस्ट वी कैन पुट हियर द इमेज. यू कैन सी द ग्रीन इमेज. यहाँ पे आप कोई भी पिक्चर अपलोड कर सकते हैं और उस पिक्चर के अकॉर्डिंग यहाँ पे जी कोड फाइल, एसटीएल फाइल खुद ही जनरेट हो जाएगी. अगर आपको क्या करना है कि आपका कोई पूरा कंस्ट्रक्ट ना क्रिएट करके आपको एक पार्ट ही क्रिएट करना है, तो आप जस्ट उस पार्ट पे क्लिक करेंगे और आपको एक लाइन अंडरलाइन होगी उधर जी कोड फाइल से कि आप उस लाइन को एडिट कीजिए. तो आपका ये वाला जो पार्ट है वो चेंज हो जाएगा. तो आप सिर्फ पूरी फाइल को एडिट ना करके एक लाइन को एडिट कर सकते हैं, जिसके थ्रू आप अपना जो कंस्ट्रक्ट है उसके स्ट्रक्चर को चेंज कर सकते हैं. तो यहाँ पे आपको कोई एक्सपर्टीज की जरूरत नहीं होगी कि आपको कोडिंग आनी चाहिए. आप इसमें किसी भी स्ट्रक्चर का या फोटोग्राफ आप इसमें डाल के जी कोड फाइल जनरेट कर सकते हैं.
Okay, thank you so much for nice explanation. Actually, there was some misplacement of slides, video or something there. So, she wanted to ensure that no information should be missed out. After that, the talk we are here to discuss all your queries. So, yes, we are talking about this DNA Studio 4. This comes with this machine. You need to. There are several preloaded parameters, models, programs are also available in this software. You can just select and make the structure ready and you print the thing. One very important thing is being done from Cellink company. They are maintaining one repository called Bioworks, where they are collecting information throughout the world. Whatever the organs structure has been printed, You simply go and check it, and if it is of your use, you can take it. Or for example, you can pick that structure. I mean the G code file or STL file. You can do modification from that, and because they are maintaining their all languages, G code and STL. So you can simply select the structure and you change the thing. So you whatever material, biomaterial, whatever the 3D printed structure available in this world, they are making repositories. So they are making life easy. So you can collect all information and you design your experiment. So after discuss about the 3D model, how to select the model, how to design the 3D model, the next one is for this choosing bio-ink and biomaterial. So for good biomaterial, there are certain prerequisites that, for example, first, if you are thinking of bio-ink preparation, you have to simply take biomaterial and cell suspension and you just prepare biomaterial. So biomaterial needs to qualify two criteria, that it should have good bioprinting fidelity. I mean, it should not spill out while printing. You see the structure. You see, if this is good biomaterial, it will be printing like this, and if it is not good fidelity, it will spill like this. Is it clear? No voice, means like it's not clear. Am I audible to? Please, backside person. So is it clear to you? I wanted to highlight these things, that if your biomaterial is good quality, then it will give the good shape. If it is not good quality, it will be spilling like that. So you have to consider which biomaterial is needed for your application. For example, if you want to print soft structure, if you want to print hard structure, so depending upon your application, you need to select the biomaterial. Something like some biomaterial need high temperature, some biomaterial needed low temperature, some biomaterial need different cross-linking. So accordingly, you need to select biomaterials. Plus, apart from it, the other criteria it should fulfill, that it should not be cytotoxic. I mean, what are the downstream applications you are taking the cell? It should be cell-friendly. So you better know which kind of cells, which kind of experiment you are planning. So based on that, you can select your biomaterial and you just proceed with. It should not be cytotoxic. So it should be cytocompatible. So this is again important slide. Master students, please have a look on this thing. This is very important thing. What makes bioink? For bioinks, the polymer is definitely the major component. After that, you have to add thickness. After that, you need some bioactive components, for example, generating free radical or something like that. On top of it, you need to add some complementary components. For example, you need to add peptides, growth factors, minerals for cell growth. Sometimes you need complementary components. Sometimes you need some photoinitiator to provide free radicals or something to initiate the process. So I will, and again, CellLink had one complete list of biomaterial. You can go and check Advanced Biometrics website where a complete list of biomaterials are available. You can explore it, and where all details, including percent and for what application you can use. There are application notes also. So based on that, you can select your biomaterial and you can define. You can proceed with your downstream application. So two kind of polymers you can use. One is natural, one is modified. So of course, most of the cases, if you go with the natural polymer, it might not be suitable for you. So it needs to be modified slightly. So for example, if you talk about the natural polymer, You can take alginate, hyaluronic acid, chitosan, collagen. If you go with some synthetic modification, for example, with UV modified like Gelma, Hama, Colmar, these are methyl acrylate modified, so you can select accordingly and you can proceed. So again, this is just a compilation of this previous. This is summary slide kind of. You can check like as a photo initiator. I told light. This is blue light 405. This is buffer. Sometimes you need dilution agent, like some for reconstruction purpose. You can add some dilution agent and you can make your polymer and biomaterial ready. Growth factors I mentioned RGD, laminin, tricalcium phosphate, hydroxyapatite. You can take it and you just do your application. This slide is again very important. Like in earlier video, we have seen that if you are curing with arginate calcium chloride, the structure was more stable. If you don't cure, once you print it, you cannot use downstream application. So there are 4 types of methods how you cure in the structure. I mean, once you printed the structure, after that you need to cure as per your chemical nature, polymer nature. So either you can use ionic solution like arginate calcium chloride base, you can do that. Sometimes you need to simply use UV light like Gelma, crossing Gelma network, where you can use UV light. Sometimes you need temperature based, like glycogen and this gelatin. They are just setting their structure by simply increasing or decreasing the temperature. So you just tweak the temperature and you make the your printed structure pure by simply by changing the temperature. Sometimes you need enzymatic. For example, fibrinogen to fibrin polymer conversion, you need thrombin. So here, by using that enzyme, you just make the crossing the structure. The summary, here I wanted to explain simple that whenever you just print the structure, depending upon your biological material, you need to cure the structure so that your structure should be stable. So you need to follow this simple steps. So you better know based on your experiment which curing process is suitable for you. So you plan your experiment accordingly. We have this cell link explore. I think probably today's one we are going to use this. Yes, so we have the cell link explore. It's already arginate based binding, so we will print some model by using this in second half. So here cross linker is calcium chloride, which generates ionic bond. So like that, we have complete range of biomaterials cross linkers. So just to mention a few, this is one example. Having discussed about designing the structure, can you recall? I would like to hear from you guys that what were the steps for bioprinting. How many steps we followed? For example, we designed the structure. What was the next step?
Select the biomaterial.
The next step? The next step?
Curing.
The next step?
Incubation.
After that, ultimately, the downstream analysis, whatever you are repeating with 2D things, you can just go with the same thing. Please ask questions if you would like to, else I will think that everyone has slept, no one is listening like that. So this will give energy to me while talking. So you can ask a question. Otherwise, I will be also monotonous. Simply, I will do slide reading and I will also not understand what we should be delivered, because that's what sometimes we will try to summarize this thing also, whatever discussed earlier. So yes, great. So in the beginning slide, what I discussed about, okay, this is again I am asking question. In the beginning slide, what I discussed, what kind of portfolio cell link is offering, like for the bioprinter. Can anyone tell? I discussed that we have two types of portfolio. One is the pressure based, the pneumatic based, the other is light based. Is it clear? This will give energy. I think otherwise I will be monotonous. Please reply. This will give definitely. And by doing so, I will also understand that how much I am able to explain it. So, we have two types of printers. As I discussed that one is the extrusion based, I mean the pressure based, and another is light based. So, we will try to cover one by one. We have dedicated instruments. So there are from very beginning we have incredible and incredible plus. It's a very old model. Just to show, I have included here where the print head were only one print head. So it is the output was less. So the technology there evolved. So we have Bio X and Bio X6. Here major difference, keep in mind that in BioX we have exchangeable print heads, 3 exchangeable print heads in BioX. In BioX 6 different exchangeable print heads. What is the need of this exchangeable print head? For example, if we would like to print skin-like structure, skin is very complex structure. For example, we need to print hypodermis, epidermis, hair follicles, blood circulation, blood vessels, related things. So the major USP of this, I will explain in going slide more. The major USP of this Bio6 machine is coaxial printing is possible. I mean to say, if you want to see like a structure, so at the same time, you can print the outer sheath and the inner core like a structure. That is the major advantage of this machine, that blood vessel like a structure you can print on the go. Plus, Bio6 material like 6 exchangeable print head, we can use 6 different type of biomaterial. Also, in each print head we can use different print head. For example, in something like we can add gelatin, in some we can add temperature sensitive, some we need some for example printing hair follicle for skin like structure. You need to control the volume. It should be very fine, it should be very thin. So accordingly you can use that print head, sealing pump print head, and you can control that process. So it is highly doable. I mean, in all 6 different print head you can feel there is one slide I will show. You can use, they have a study have used 6 different type of biomaterial all together, and how they have printed the skin like a structure. This is very wonderful thing. So we will we'll see in the coming slide. Yes. So again, this model will keep on repeating to remind you that we have discussed the 3D model, we have discussed the bio. Now we are at the stage of this machine stage. This machine we are looking. So in this we will first we will discuss about the pressure based machine. In the next we will go with the light based, where we have two portfolio. The Moga X is high throughput, which is in latest technology. Before that we were having Lumen X, where throughput was less. So you see the extrusion. Is it clear what is pneumatic or extrusion based? Everyone is clear? Pneumatic or extrusion based? No one. Everyone is clear about pneumatic or extrusion based printer? Here simply we have to apply pressure. After filling the biomaterial, then you can use the material will come out from nozzle by applying pressure. Whereas in light based thing, the material will be, the structure will be printed based on the area of light. I will try to show in the coming slides also. By area of light, from bottom to top across, light, amount of light passes, the structure will be printed in layer by layer. So these are the major differences. That one is pressure based, one is light based. There are, this is typical machine which is in display. So this machine is very compact inside. You can see it's hardly 2 by 2 feet, something maybe like that. It is having HEPA filter technology, so you need not to worry about your sterilization. HEPA filter is there, so this machine is able to make the chamber clean with the help of HEPA filter. It's already installed there. You can clean chamber technology is there. Plus, this is having multiple light storage system. You see, 365, 405, 475 and 520 nanometer you will lay it there in the bottom. I think Simani will show while the demo will show that we have in this machine UV lights are also there, so you can use as per demand. Depending upon biomaterial, depending on application, you can try it. It has temperature controlled print bed. This is print bed. This is print bed where the temperature can range from 4 to 65 degrees. It has peltier which will make the temperature cool or heat very faster. So depends. Sometimes you need 37 degrees to seat the cell. Sometimes you need 4 degrees to make cells more stable. So depending upon your experimental plan, you can play it. So this machine is capable of handling. This is very handy. You can see the simple plug and play machine. Simple plug and play, and this machine can handle up to 700 kilopascal pressure. You imagine 700 kilopascal. Inbuilt pressure is 200 kilopascal. Apart from that, you can add external pressure, but in most of the biological processes, hardly you need more than 50 kilopascal. 99% of the structure is printable in the 50-60 kilopascal range. And here, again, one main thing that once you design the program, you decided the pressure, you decided the velocity, and you decided everything. On the go, you can change the pressure and velocity. We will try to show that part also. It's not that while you need to wait till the last. Means while printing you need not to wait till the structure. While printing, if you come to know, no, this pressure or this is not making proper shape, on the go simply you need to tweak the parameter. I mean pressure, velocity, and you can see the output. So you need not to wait till last. That okay, after printing the structure I will see. If you understand that no, by changing the pressure, it is making the structure more crystal clear or something, so you can change it. This is all doable, and of course it is so handy. Hardly 7 touch screen, simple touch, and you just change the program. We have BioX 6 where there are separate laptop comes. You can design and change the parameter in that machine. So, having discussed about this machine, I think here I was discussing about print heads. We have exchangeable print head, that is one thing. We have a different type of print heads. See, this is pneumatic print head. We will be using this print head today, pneumatic print head. We have temperature controlled print head which goes from 4 to 65 degrees. I think it is in display. We have electromagnetic droplet printer (EMD), which is kind of like inkjet printing. For example, if you have some very thin things, you just print in inkjet. I think one video is there. Probably I will display so you will understand how every. I think in next slide this is there. After explaining, I will run that video. So in that it's simple, just you need to print like inkjet and you print your structure. We have thermoplastic printer. I think it is also in display, which you imagine the temperature and it can reach up to 250 degrees. So you can, depending upon biomaterial, you can increase or decrease the temperature. We have series pump printhead where you can change the, you can have precise control over volume. So you can easily distance or control your volume by using that series pump printhead. Along with we have total curing tool head also where 365, 405, 450, 485 and 520 nanometer guidelines are there. You can use it. Can you run this video? You see how this different printhead. It's a pneumatic printhead. This is a petrol plate and this is 96%. You see the thermoplastic printhead how it's going. I told the EMD printhead, the inkjet kind of thing. You see how it's going. Yeah, it's done. One very interesting example I would like to explain. For example, we are having time or we are running out of time. Okay?
Yes, sir, we are.
We have time, no issue. Because based on that I will try to. So I have taken some example in slides where we'll try to show this 3D printing technology applications. Having discussed about the biomaterial, hardware, software, we will now come into some examples. So as one example, I am going to show ear printing. Do you think the printing of ear is possible?
No.
Backside.
No.
It's not possible.
No.
Here I will show that how this technology I will use to print ear-like structure, the pinna-like structure. So we have some video. If it will play, I will show you. So ear structure is so soft that while printing you need some support. Otherwise, like say, it's mostly cartilage kind of thing. So you need some support. So what happens is that while printing, we need to provide some sacrificial link that will provide support. While printing, this will provide support like a structure. The moment you print it, after querying, we will remove all the sacrificial link and your final ear structure will be ready. So I will show video from that part. Some overhang like structure, you see in this slide, overhang structure. While printing, you have to design overhang structure. How to print the overhang structure? Need support during printing before curing. So you need to support material. Once it is printed, you can remove the support after curing. Please make an example. This printing a small ear shape with supporting. In front of your eyes, I will run the video how they were able to successfully print the structure, ear structure. You will see the ear structure, this is the support structure. See, after printing this support, they started printing this structure by this pneumatic print head. After that, I think please you can you see the once they have printed the base. The next video, please. You see. Is it clear? Once it is printed, I think you see the once the structure is printed, you need to remove the support part. You need to cure it. After that, your ear-like structure will be ready. So will we run the video again for ear-like structure or okay? Clear?
Yes, sir.
So you see, I mean to say, I want to explain that any structure, either hard or soft, if you would like to print, there are certain parameters you can apply it. You if you need some overhang structure where you can select support material, sacrifice a link, and you can print the structure. Once your structure has been printed, you can. This is again summary slide. You just have a look on this slide. This is post-print process and analysis. Once your structure is printed, after that you can do several characterization process. For example, if you want to do for biological characterization, if you mean to go for viability assay, histological analysis, cell-cell interaction, cell-ECM interaction, that is all doable. You can repeat all the down the line analysis what you were doing with the 2D things. After that, you can use different characterization method also, NMR, SDS related things, Western blot, or you can do. So, there is only difference if the structure is getting printed. Once structure is printed, after that you can do all analysis through the chemical analysis, mechanical analysis, NMR, and all the chemical treatment, cell seeding. So, after printing, all different type of characterization process, biological post-printing operations, like you can seed cell. If you need after printing, you want to add cell, simply you add cells and you do your downstream application. So everything is doable after 3D printing. Once a structure is printed, you can do all downstream application to characterize your 3D model that okay, this model has worked or not. For example, if you want to try with pharmaceutical drug testing or something, you design some steroid or some model, some liver model or lung model, you just apply your drug and you do the downstream application like inflammation assay, drug dose assay, even microscopy, you can do all the analysis. This is again important slide. It's not that this printing is possible for only animal thing. This printing is possible for bacteria as well as plant also. In bacteria, one group they have shown that they have checked for acetate production. So they used Aspermosa water cells and the biomaterial from cell leaves they used alginate and nanofibrillar cellulose they have used. They printed the bacteria things. They have checked for this bacteria is known for acetate production. So the 3D printed bacteria where it was found that 3 to 4 times increase in acetate production by object. So you see the utility of this technique, not only human therapeutic thing, in bacterial thing also you can apply it. In plant sciences, for studying the stem cells, there are few reports, but the studies have started coming also. In the plant also they have checked for stem cells and their growth related things. So, in the soft robotics also, in the beginning of talk, sir, I mentioned that it were used in robotic. So you see for magnetic nanoparticles, carbon nanotubes, thermal responsive things, they have used different type of bioprinted things and they have applied it. So here we'll try to understand what are the challenges tissue engineering. If we go with the tissue engineering, what are the challenges or what are the things that needs to be considered for engineering 3D models. So what the challenges tissue engineering, if we start with 2D culture, the cell will be like monolayer. After that, if we need to print the 3D tissue and organ, so once the cell layer starts getting deposited, the cell layer will be thick. So it is highly possible that the top layer will get proper oxygen nutrient, but bottom layer will not get proper oxygen and nutrient. Ultimately, what will happen? Cell will die, or there will be not proper uniform circulation of oxygen nutrients. So if somehow cell is getting disturbed, oxygen supply is disturbed, then your whole ETS is disturbed. So how you can imagine that you have printed model or the organ, and if it is 2D, it will behave as like a physiological environment. So for that you need while printing the structure, while doing the analysis in tissue engineering, you have to have proper vascularization arrangement, and which is very easily doable with our machine. I will try to. Again, I will ask one question. If you are not asking, I am asking question. That what I have shown with the Bio 6. Please tell me from back side.
6 acceptable printed.
Hello. I request to all students and faculty members, if you have any query, you can ask the question in between the session. There is no issue.
Otherwise, I will think that everyone is understanding everything or everyone is understanding nothing. This gives two messages that I am sure it's not that even I have taken a long time to understand the process. So in one time, if it is. Yes, please, sir.
Very nice presentation, sir. Out of the three, I have one question. This is about the drug delivery system. If we want to prepare a tablet or any kind of a capsule, so you already demonstrated that alginate we can use as a choice of polymer. But apart from this, can we use the CMC or HPMC kind of a polymer which are normally used in the tableting or the capsules? Or if yes, so in which form? Whether the solution form, as you told, this is the ink, or as a solid form?
Again, that's a very good question. As I was discussing in the morning, that means I cannot decide at that what kind of you better know. That as an example, we have shown that we print the structure based on alginate because where all percent you know that what percent of alginate, what are the pore size, what are the things you wanted to study. If the other chemical like hydrogen, collagen, you are just checking, so you know the chemical structure of your drug. So you know the pore size of your drug, you know the moment, like if it passes to membrane or how it works, means how it goes inside the cell or what are the properties you are looking for. So based on that, you need to select. Even the biomaterial, like you can combine biomaterial also.
That is okay, sir. Means my question is, apart from this alginate or nitrogen, can we use the normally used polymer like HPMC, hydroxypropyl methyl cellulose or CMC or the lactose? क्या हम यूज कर सकते हैं?
ये बिल्कुल पॉसिबल है, आप यूज कर सकते हो। अगर आपका डिपेंड करता है कि आपको कौन सा ड्रग, मेरा वही मैं आप काउंटर ऑफ इट व्हाट आई एक्सप्लेन, ट्राई टू एम ट्राइंग टू एक्सप्लेन, जो आपका ड्रग है यू बेटर नो उस ड्रग का मोड ऑफ एक्शन, बिकॉज आई एम नॉट द फार्मासिस्ट, यू बेटर नो मतलब कि जैसे आप उसको उस ड्रग की एक्टिविटी के लिए मतलब क्या प्रोफाइल चाहिए, क्या वो ड्रग सेल के मेम्ब्रेन के साथ या फिर मतलब वो कैसे पास करेगा, सेल के साइड अंदर कैसे जाएगा, व्हाट काइंड ऑफ पोर साइज यू नीड, व्हाट काइंड ऑफ फॉर एग्जांपल अगर उसके साथ सेल में कोई सिग्नलिंग स्टार्ट होती है तो क्या कौन सा बायोमेटेरियल विल बी, व्हिच बायोमेटेरियल विल बी मोर क्लोज टू योर सेल सिग्नलिंग, या फिर जो डाउन द लाइन कोई इन्फ्लेमेशन रिस्पांस या फिर कौन सा उसके क्लोज होगा, बिकॉज देयर आर मोर देन 100 200 बायोमेटेरियल्स which are being used depending upon application or even mixture of biomaterials. So based on your drug types, you can take a call to which type of biomaterial. There are literature and report on advanced biometrics. We have one website from our company where you can go and check that is there, because I am not recalling the application exactly, but there are notes which suggest that for which type of drug, which type of biomaterial you can use. You can go and check our website where you can get clear-cut idea that yes, for like simple cellulose or nanocellulose or something, or calcium hydroxyapatite, or you can use for your downstream application, or our expert will explain you that if you have some drug, so you want some suggestion from us, like you are asking. Right now I am not in position to explain that for which drug, what kind of biomaterial is someone has done or some study, because there are so many R&D going on. Definitely, I am sure there will be some reply for your question कि वो टीम के पास होगा, but I am not in position to explain right now कि किस ड्रग के लिए कौन सा, there are list मे भी on website पे होगा या फिर उनके R&D टीम के पास होगा. If you have query, just mail me. I will share my mail ID. so that I can come up with the solution that no, sir, for this pharma, this product, this drug, you can use this biomaterial as a cocktail. And you just print that structure. Again, it depends on which kind of a structure. If you are taking on the skin or maybe toxicity, SC or something, you can do that.
Thank you very much. And my second question is, since we are having a very accessibility to any animal organ, so what is the necessity of this machine? Because if I want to conduct any experiment on the skin, so I just need to bring the approval from the IC and can perform that experiment.
I agree, but there are certain Western countries where people are going to ban animal system. First, in reply to your question, another thing, if you have animal system, for example, you have to grow hamster, rat, mice, whatever. So they have particular lifespan, so that you need to wait till that. But today itself, you can print the structure. You know the parameter of your that liver thing. You just print the structure. You need not to wait for 30 days or 45 days or something. You just print the structure. After that, you seed your cell. While printing or after printing, you can see the drug effect. The other thing, like for example, equal again the same. I was discussing with Siddharth also in modeling. This is very important thing that there are wonderful outputs from the drugs which were doing in vitro testing and all these things. The moment it goes to animal system, it's getting failed. Why? Rat genome, mice genome, 99.9% you can think that okay, it's similar, but those 0.1% differences will make huge impact in the outcome of protein. So I am emphasizing that in place of your animal model, if you will take, for example, there is one company, I think I will cover that example in like one company called Classic. They have checked for pancreatic cancer. They have taken patient-specific cells. That's what I am trying to emphasize. Patient-specific cells. For example, most of the cancer patients will not die with the cancer. At the last, the radiotherapy or chemotherapy, the overdose or underdose, they die ultimately. Some thing like it is sometimes you are giving overdose or sometimes underdose. So if you take that cell from that particular patient and you define the chemotherapeutic drug dosage, because it will again vary from patient to patient. I will for your just that I am counter of your question. I have something more interesting to explain. I was discussing in AIMS Dental Science, New Delhi. They wanted to prepare implants for cleft palate. Cleft palate, everyone knows, cleft palate is the kata wala out. What the kate wale hote hain. This is very common. Its frequency, I think, in 10,000 is stay. So it needs to be designed person specific. It's not that आपने एक इम्प्लांट बना लिया आप सब में ट्रांसप्लांट कर देंगे. Getting my point what I'm trying to say. So 3D printing का necessity मतलब हर एक चीजों के बेनिफिट और नुकसान हो सकते हैं, तो वहाँ पे भी 3D प्रिंटिंग आप हेल्प कर सकता है कि किसी पर्सन का जैसे फॉर एग्जांपल किसी एक लेफ्ट प्लेट है, आपने वहाँ से सीटी से उसकी इमेज ली है, अंदर से इमेज ले लिया, तो उसके अकॉर्डिंग यू आर डिस्कसिंग अबाउट दैट व्हाट व्हेन आई ट्राइंग टू एक्सप्लेन द एचटीएल एंड जी कोड इंपॉर्टेंट. Might be this is first time you are listening this word, maybe someone is knowing from beginning also. So वो हम बताना चाह रहे हैं कि simply by changing one line, you can change the dimension. Like some person having 3.5 mm, some person having 3.6 mm. By simply picking the, you just print the person specific construct. Getting my point what I'm trying to say. जैसे आपने जेनरिक ड्रग जनरलाइज ड्रग टेस्टिंग कर लिया, लेकिन अगर आपके पास कोई पेशेंट का स्पेसिफिक जैसे कीमोथेरेपी जैसे कार्सिनोटिक जैसे कमर्शियलाइज, तो अगर उसका उस पर्सन स्पेसिफिक अगर हम वैसे करें तो दैट्स व्हाट पीपल आर एडवोकेटिंग कि नहीं आप हायर लेवल 3D प्रिंटिंग आप यूज करें. ये विल बी बेटर सूटेड. आप बोल रहे हैं ना कि हमारे पास तो बहुत सारे एनिमल है, हमारे पास तो मैं हजारों सैक्रिफाइस कर सकता हूँ. हो सकता है कि 10 साल के बाद लेबर किसी ने बोला गवर्नमेंट ऑफ इंडिया आती है तो कि कोई एक्टिविस्ट आते हैं और बोलते हैं कि नहीं आपको एनिमल को आप नहीं कर सकते. वी आर लिविंग इन द कंट्री लाइक मैं उससे ज़्यादा नहीं चाहूँ बोल करता है. बट द पॉइंट इज कि कोई अगर आता है, कोई आंदोलन आ जाता है, कोई मूवमेंट आ जाता है, दे स्टार्ट थिंकिंग नो कि आप चूहे मत मारिए, मतलब चूहों को सेक्रीफाइस मत करिए अपने साथ करो, तो अब आप कहाँ जाएंगे? अगर आपके पास ये तैयार है, ये प्रोटोकॉल तैयार है, देन यू विल बी बेटर, आपका रिसर्च नहीं होगा और इसके वजह से आप पता है सर, आई एम नॉट श्योर कि आपका एनिमल का हैंडलिंग और उसका बहुत कॉस्टली आते हैं, जैसा मैंने सुना है, प्राइस आई डोंट नो, बट मैंने सुना कि जो बाहर से आते हैं क्लियर साइंटिस्ट बताते हैं काफी कॉस्टली आते हैं, उनका ब्रीडिंग मतलब उनका जो को मेंटेन करना, कंटामिनेशन से बचाना, उसके लिए आपको एक स्टाफ चाहिए, सुबह शाम देखेगा क्या इन्फेक्शन हुआ क्या नहीं हुआ, फिर वो दूसरे को कर अनइंटेंशनली हुआ राइडिंग सा करिएगा। लेकिन यहाँ पे जब आपका जो प्रिंटेड मॉडल हो जाएगा तो आपका जो मतलब वहाँ पे वो सारे भी आप अवॉइड कर सकते हैं, राइट? प्रोबेबली आई थिंक थोड़ा सा मैं 10% आपके क्वेश्चन का आंसर करने में मतलब कामयाब रहा। After the talk, we'll try to discuss more. So, there are the challenges in the tissue engineering. I discussed about the thing. System needs to deliver oxygen and nutrient. Hi, Moga. Yes. This, please attend on this slide. Actually, the yeah. Yes, sir. Yes, so इसमें the moment you print, you wanted to print the tissue engineering from top of it, like there will be gradient of oxygen and nutrient. I was discussing about. That top layer cell will get more oxygen, more nutrient, but bottom cell will get bottom layer there will be more dead cell. So if we have proper vascularization, sorry. So if we have proper vascularization, then cell will get proper nutrient, proper oxygen supply and everything along with so that you can mimic the exact physiological environment what cells are getting inside. Like in hand. बात करने का ये दिस इज अगेन इम्पोर्टेंट स्लाइड बिकॉज़ मुझे लग रहा है कि शिक्षक लोग बिल्कुल मोनोटोनस हो जाते हैं, तो मुझे लगता है कि मैं ऐसे स्लाइड एडिट कर रहा हूं काइंड ऑफ. So see this is an interesting thing. The creating channel-like structure with extrusion-based printer. If you want to make channel-like structure, we discussed the importance of channel-like structure. So we need to create channel-like structure. So what are the base steps involved? So first we print the base. After that we print the channel structure with sacrificial. Sacrificial in, for example, folic acid. You just print the structure. After that, printing the top. While curing, we will remove the things so that there will be channel-like structure. I will show in the next slide. You see the fill the rest with the base material and cross-link it. After remove the here we need to add video just I am talking about. Remove the sacrificial link. The channel-like structure is prepared. The top panel. This one here. This one video. First, please run this video. See. This structure has been printed after this thing. Please run this video. Excuse me. Please run this video. This one. See the structure, the channel like a structure is printed. You see the moment, the next one, the right of it, you can run that video clip. You see. Once you inject the material, it is going in things means the structure has been printed. So this is possible with this machine also. The channel like structure printing is possible with this machine also. If you want to go with like skin like a structure or high level structure or complex structure, for that you use bioelectrics. But simple channel like a structure is possible with bioelectrics. Now the important feature of BioX 6 comes. It has 6, as I mentioned earlier, it has 6 actionable printhead. It has coaxial printing platform. It comes with tablet. Here we have a touch screen. In that it comes with tablet. Easy to use coaxial model printing. I was discussing about that in that thing. So easy to use coaxial printing is possible. See the outer sheet and the inner core is print. It is possible to print together. Please run this video. You see how it's possible. See the beauty of coaxial printing, that your outer sheet and inner sheet with two different type of raw material, it is getting printed. So once creating a permeable channel, how like a structure, because most of the structure need like if you have you want to work with complex structure, you have to have proper vasculation. So in that case, you can print the sheet. Again, I am coming to the very interesting thing. So, उस slide में आप लोग जग गए please. So, we go here channel like structure under we printed a coaxial printing. After that, removal of sacrifice and the outer left behind. You see the skin model. This is very important slide. This is the skin model. Like skin, a typical skin structure. Epidermis, dermis, hypodermis, like epidermis, you need to add keratinocytes, melanocytes, hair follicle, blood vessels. So you need to design your experiment that your program and parameter should be able to print epidermis, hypodermis, hair follicles, blood vessels. Getting my point? So you have to design experiment and select the parameters accordingly. Which biomaterial is close to epidermis, which biomaterial is close to hypodermis, which biomaterial is close to hair follicles. So it all completed. This is one program. In the next slide there is one all biomaterial are shown. You need to print the dermis, hypodermis, epidermis, like that. This is very summary slide where the skin structure has been printed. You see, you see the thing that for printing BioX 6 machine has been used, where 6 different print heads present. There is 1, 2, 3, 4, 5, 6. You start with the first, like in first pneumatic coaxial model used. The second step was also print with pneumatic. They have used gel X-ray and UV cell for printing this sheet-like structure. They have used chloronic acid for sacrificing, so that will be removed. After that, for hollow-like structure, 130 ready. After that, they have used temperature controlled print head to creating the base, gelatin based barring plus free adipocyte cells. In the next step, they have used temperature controlled head again, with gelatin based barring plus fibroblast plus leukemic cells. After that, they have used the EMD. EMD inkjet print head and they use gelatin based body plus melanocytes they have used and they printed. In the next step they have used syringe pump where volume needs to be controlled and they printed again they have used gelatin based mostly. So gelatin based body they have used hair follicle cells. So you see the complex structure of this skin. How is it possible to build complex structure of skin? So these are some few examples where 3D printing for skin application have been used. For example, in wound healing they are used skin graft, skin sensor. In drug delivery they use digital modeling and of course cosmetic testing. So there are lot many examples, not single. That has seen there are wide, so many studies have been reported and people are working in this field. So I've already discussed about this in immunized skin model. Like the world first 3D bioprinted immunity model for screening drugs and ingredients for normal and inflamed skin. The CGI biotech they have done. So the carcinogenic, I have also covered this example already in the beginning. As example I have given. They have taken the tissue samples and they printed and they have done the drug testing. Again, this is very important example where for viral influenza like study they have checked the thing. Here, one thing very interesting, they have used 3-layered model. Like 3-layered model structure where in the first layer they have used the bar-ring one. In the second layer, they have used, in the third layer, they have used bar-ring 3. In the second layer, they have used the bar-ring 2. With 1 mm thickness, they have made it. And for biomaterial, they have used alginate, gelatin, collagen, calcium sulfate. They have used the A549 non-carcinoma cell. They have used the normal cell, NHLF-B and THP1. And they have printed the structure. Incubated it, cured it, after that incubated with the influenza virus and they have checked the viral infection, how it moves. So they have checked for 3 things, downstream analysis, they have done with the cell viability, they have checked the metabolic activity, they have checked for cell morphology and protein expression analysis. So you see the inflammatory response also they have checked and they have checked for viral infection. So I will just go to some slides. So what are the model of like cultured cells in 3D? I discussed multiple cells like they used epithelial cancer cells, they used fibroblasts, they have used leukemia cells, they have used LPS stimulation, viral stimulation study, various analysis, they have done cell viability, metabolic activity, related things. And the key findings were the 3D printed cells were viability was up to 35 days. There were more viability in the 3D printed things as compared to 2D. Plus, the more better immune response observed and the inhibition of virus growth with antiviral drugs were also increasing drug dependent model. So you can understand, you can imagine how this 3D printing, 3D printed model is helping. By clearly, you can see 3D. Again, here I want to give one important message that 3D printed cells they are having more viability. Like why? From back side, why it is more viability 3D printed cells? Because the bottom layers and all the more focus circulation of something. That's what I am saying. Everybody is either feeling hungry or something. So we'll close in 5-7 minutes. So, what are the advantages of 3D bioprinting? For example, in pharma company, if you want to fabricate it, you need more construct. So, if you go with manual thing, there will be different size. In some smaller, some bigger, something. And if you go with 3D printing, you will have throughput, high throughput. It is in more than 100 models and with equal size is printing possible in 2 hours. So, you see the utility of this technique. That's what I wanted to In every section, I'm just emphasizing that how 3D printing can help for everyone. Like in biofabrication also, if you are going high throughput, you can just apply this and you will have better output, better control over that. For example, if you don't have your some construct is bigger, some construct is smaller, your drag will be different, the response will be different. Here more, you have more control over the process. This is again an important thing, the cornea model. They have printed 3D printing cornea astronomical. So for that, what they have done, they have just designed a 3D model. They have taken human cornea, molded the 3D structure, and just framed the structure, and just printed the cornea like the structure. They have used collagen, alginate, corneal granulocytes, and they have printed the things. 3D printing and incubated and printed cornea construct. In India, Pandorum technology already started the commercialization. So you see, there are success stories also. It's not that success stories are not there. In India, yes, I'm not talking about outside. People are making this thing. We are discussing in several issues. People are using this technology, and who is not using, they would like to use it. So, as provided like a structure for creating heart-like model, they have used they printed cells, they made astrocytes, and for printing in vitro heart model. I will just try to summarize in 5 minutes. They used astrocyte models, fusion of 3 cardiac cells. Astrocytes, they created cardiac myocytes, fibroblasts with the glial cells, and they used again alginate gelatin-based binding. And the key findings were the high cell viability, the similar story. Here they are also able to see the more viability, better angiogenesis, the astrocyte fusion and astrocyte beating also. Means like this is close to natural thing. So you see the utility of this technique. Again, the neuroblastoma model they have used. I think sir is working on neuroscience probably, so this might help you. The neuroblastoma model, glioblastoma model also they have used with the pneumatic printing. So they have used BioX, BioInk, they have used Gelma and Hexacure. Support bar, actually again, I was talking about support structure. Here they need support bar. In that bar, they need certain particular temperature, certain chemical environment to print the structure. So they use carbocol and they printed the structure. They use the human cells and the neuroblastoma cells as provided, and human derived epithelial lines IL-5, and they printed this neuroblastoma model. So you see, there are several success stories for this thing. This is again the example for printing the structure. You need to provide support bar so that your structure or construct should not get damaged. It gets proper support while printing. Coming to the latest technology, which is high throughput, which is Moga X. Where the digital light printing is possible, it better resolution, and it works in high throughput manner. Again, there is no issue of pasteurization. Again, it can take control of sterility. And here, the pharma company, or if you are going for long high throughput part, you just use this technique and direct involvement. So 96 well, this machine I was training also. For Moga X, we can automate it, and you can go. So again, there is a video, but I will be running out of time, so I will make the escape. Creating a small channel-like structure. This part will just in 2-3 minutes I will summarize. We have discussed about this the pneumatic based. So there is again, it is all channel-like structure printing possible with light-based also. Light-based. Simply by passing the light from the bottom, the layer to layer structure, then by automatically the channel-like structure will be created. I will show some figures. We discussed about this extrusion based. We will sell like human X and Moga X 2 portfolio. We have here you can just print the construct photo. You can use the photo durable material. From projector light will go and the print smaller and more complex structure. This is the space. This first layer printing. These 3 panels show one is side view, one is the poly view, and one is 3D view. All are similar thing, but in the different how from side how it will look, from the 2D cross section how it looks, from 3D perspective how it looks. You see, the first layer from light is created. After that, the channel from the right side of the channel like a structure will be created. The second layer from bottom is getting created. The next third layer printed. So you will see this inside view and it is 3D printed structure. This side will create the channel like a structure. Getting my point? Is it clear? So printing live cell is also possible with this thing. You can print the live cell by just taking control of temperature and you can print it. There is a video, I think you can please run this video, this one. First we put the wire material, start the print. This is for the luminance, you see. Is the structure ready through the light based technology. There the structure where with the pressure vector model. Heart model, based on that they have created heart model also, where they have taken embryonic heart and fetal heart. They have created the structure based on light. They have seeded with liver cells, and the channel like structure was able to perfuse the material. So we have solutions for this high throughput. I discussed about the Moga. So certainly 3D bioprinting has wide range of applications. For example, design flexibility. You can design organ-specific model. You have different products like tissue-specific, patches, implants, and a wide range of models like organ model, living model, food applications of robotics. We have broad bio selections like gelatin, alginate, collagen, chitosan. I was talking about advanced biometric. We have different additives. You can use antibiotics, bioactive molecules like dynamic particles, and various cell types also cell lines, primary cells, in embryonic stem cells you can use. So we have the whole portfolio, starting from hardware to software, chemical to consumables to plasticware. All solutions are there. You need not to waste time in optimizing that part. You just let us know. We will provide which step and which material is best suited for your purpose. So this is just summary. I mentioned already. I covered about this. Extruder-based biodistance and light-based biomaterials. So we have bioins available already. Just check our website. So you have more than hundreds of biomaterials available. So that's all. Thank you.
Thank you, Dr. Santosh, for this wonderful information and इस टेक्नोलॉजी को हमें देखने का मौका मिला है. मैं आप सभी को एक बात बताना चाहता हूँ. This instrument is manufactured in Sweden and this is the single one instrument that is available in India for demo purpose. और सर आपने सबसे अच्छी चीज़ हमें स्लाइड्स में ये लगी कि आपने जितने भी एविडेंस दिए यहाँ पे उन हर एविडेंस में एक सेलेक्टिव रेफरेंस BOI नंबर रेफर था और विद रेफरेंस आपने बात करी. आपने हमारी फार्मास्यूटिकल रिसर्च को, फार्मास्यूटिकल मैन्युफैक्चरिंग को और फार्मास्यूटिकल जो हमारे एसोसिएटेड फील्ड्स हैं उसमें हर पहलू को छूने की कोशिश की. चाहे वो ड्रग डिस्कवरी है, चाहे वो अल्टरनेटिव टू एनिमल मॉडल्स हैं, चाहे वो पॉलीमर्स हैं, हर तरीके से. और हम खुश हैं कि हमें एक नई चीज़ देखने को मिली है. वी आर एक्सपेक्टिंग दैट इन दी हाफ नेक्स्ट सेशन जो हमारे पास मिस जो शिवानी मैडम है वो डेमो देंगी. और मैं एक चीज़ और यहाँ पे ऐड ऑन करना चाहूँगा. यहाँ पे फार्मास्यूटिकल केमिस्ट्री, स्यूटिक्स, कॉलोजी हर फील्ड के बच्चे बैठे हैं. तो आपके फील्ड में जो मेथड आप यूज़ कर रहे हैं, चाहे वो स्यूटिक्स का है, फार्माकोलॉजी का है, उसमें कहीं ना कहीं इन फ्यूचर ये टेक्नोलॉजी किसी ना किसी स्टेप में आपको यूज़ करनी पड़ेगी. कहीं ना कहीं ये टेक्नोलॉजी एसोसिएट है. So I request to faculty members if there is any query you can ask. So I can pass this mic to students. Yes, sir.
Thank you, sir, for very nice presentation. Sir, I have few queries. Like, please tell about what are the basic differences between extrusion and light based? There are any biomaterial based difference that which type of biomaterial is used for particular purpose or not? Second one is as you told that we can print the live pictures also, live cell pictures also. So we can print the enzymatic interactions of cellular interaction or receptor based interaction.
I have not included that slide. There was one paper in science. They have a vector exchanger. They use vector exchanger. They printed the live cells. That slide is not there. Due to time and all audience, I have just listened. So, in reply to your first question, what are the basic differences between the extrusion based and the light based? Simple technology in the extrusion based is pressure based. We have different type of print heads. You feed your materials and by applying pressure you print it. But in the light, actually based on the light area, how much light it's getting, you decide based on your requirement. So that from bottom to top approach, first layer, the second layer, third layer, and in between the whatever the shadow is coming, it will create the channel. In the light based, earlier human X was the resolution is more, but in that case, throughput is less. Here you will have more throughput. That's what our new edition by Moga activity, which works in a high throughput manner, like that. If some pharma company or someone is having robotic manner in plain manner, so it can print directly in where based on the specified parameters. On top of it, we have already set up pre-validated protocols also. Whichever scientist should develop, you can use that, you can refer it. If you want more modification, you can reach out to us. We will discuss with our expert and we will try to explain or we will try to help it out which biomaterial, which state, which machines, which print head they should have for it. Right.
Sir, my second question was that can we print the receptor enzyme interaction in living condition with the help of
enzyme receptor interaction? I am not sure.
So,
like
we are going to print the cellular structure of
right.
So we can bring the receptor like a structure or
Receptor like a structure, I think we need to, I'm not sure, we need to check the reference set first. But I think receptor like a structure is possible. If you are aware with some report or we can check. I think receptor like a structure is possible. The interaction we are talking about, I think, I'm not sure, the interaction part. But yes, receptor like a structure is possible. So we need check references and confirm, but it is possible, I think.
Sir, my one question is that is you mentioned that we require the growth factors
Yes.
for the
Right, yes, yes.
instrument. So we need a different growth factors for
Yes, because if you are working with cardiac cells, you are working with lung cells, you are working with embryonic stem cells. So definitely for their growth, particular RGD, even I think RGD concentration needs to be different. Even if you are using RGD or laminin, even for that you need to have different concentration.
Thank you, sir. Thank you.
Thank you, sir. Any questions from student side?
We can discuss further after that also.
Definitely.
I will pass your question if I am able to answer, and otherwise I will pass your information to that so that we can.
I am a chemistry person, so it is more quite beneficial if we can print the.
Right, nice question. So, like very critical questions we are talking about the interaction. It's a very nice question. That interaction is possible. We will check and let you.
Thank you, sir, for your discussion.
Sir, my question is how we can maintain the same environment.
Pardon?
How we can maintain the same environment, sir?
Cell environment cell environment needs two things like you need particular ECM. For example, you need particular type of cell, certain specific ECM. So while printing, you can use that ECM like things and you maintain the structure. Even cell environment, I think by maintaining the temperature, while we are discussing about the growth factors and all, to maintain cell stable. we have the option to print while the printing the structure and adding the cell, or once you print the structure, after that also you can seed the cell. So after printing, if you are adding the cells, you need not to worry that you are going with all your down the cell application. But while printing the duration, during that duration you can add particular ECA, some enzymes or some growth factors, so that will maintain or maybe temperature. So for time that will try to maintain. For example, some cell needs to grow at 37 degrees. You have to select your cell types and biomaterial level. That one should not go too high, one should not go too low. So that you need to design experiment accordingly. Then you make the things possible.
Good afternoon, sir. Sir, I have one more question to ask. Sir asked that could we just make a receptor from the use of this kind of apparatus? So when we say that, are we really using the cells, like real cells, or the material that can be used, like to mimic those structural activities?
No, in that experiment, we are using direct real cells. While printing the structure, we are using, that's what I was just talking about, that while printing the structure, you can use real cells. So, if you print the structure, you are taking the real cells from human cells, and you are adding some embryonic stem cells or particular cancer cell types, or maybe a specific fibroblast or some related cells, and you are printing. So you are taking the similar things. Like you are taking similar cell types. So this is the your exactly close to physiological. That's what I was talking about. That the more the drugs, why 9 drugs out of 10 getting clinical tested? Because they are getting tested on mice models or something. But in that case, if you take direct cells, I give an example of carcinobiotech company. They have taken that particular patient sample and they have tested that particular chemotherapy treatment. So it is almost similar to your life cells. I mean, you are testing the similar thing, like which how it is going to behave inside cells. Right?
Sir, I want to ask one more thing. That is this machine is capable to, like I just want to make a skeleton muscle.
Yes.
Is this machine is capable to make that exactly similar structure?
Yes, it is, but possible you have to use different print head. We have print head thermoplastic which goes up to 250 degrees. So any biomaterial, hard biomaterial you can use, and by treating the temperature you can apply it.
And what raw material will you require for that? Only the cell culture?
Based on your experiment, you better know which cartilage equivalent or something. Right now I cannot recall, but I need to check everything in website already there. Right now I cannot recall. The similar matching biomaterial can take and just print the cartilage. Yes, it's possible in IT. There is some person, I think, Shivani, what is the name of the scientist in IT? They are working on the cartilage structure. Yes, they are making the structure. We are in touch. They are using this technology. It's possible.
The material that we are using to synthesize the cell, and after the printing, are those cells living really they are alive or not, or just mimicking the cellular integrity of structure?
I mentioned two things very clearly, that while printing the structure, you add the cell lines, or once you print the structure, you add your cells and you do the line. So you decide based on your application that which way you are going to proceed with. What if your experiment demands that while printing you need to seed the cells, you just take the cells, print the structure, and after that you do the downstream application. Otherwise, once you print the structure, make the printed structure compatible to be seeded with cells, and you will proceed with that is the difference I told you. So after 2D, you are just going down the stream application. Similarly, you go with 3D application also. The moment it's printed, after that all downstream are similar. If it is possible 2D, it is possible with 3D. Simple. If you are working with a culture cell, similar study, similar analysis, you can do that.
Good afternoon, sir. My question is, does it require any regulatory submission
For
the medical devices used in 3D printing?
I think country to country some regulatory bodies should be there. I am not very sure, but I think some regulation like maybe it might be present. I think so, because you cannot like then you cannot go directly to market and you sell artificial like 3D printed heart. Like you cannot go directly to AIMS. Oh, I have printed heart, just simply transplanting. Like there must be certain regulations how printed, what are the materials, how tested. Like you have to provide certain evidence, and like no one will accept you. You have to some biological characterization and related things. So it needs, I think, regulatory body probably.
Next question is, how can we correlate it with COVID-19?
There are so many examples, like the influenza virus example and COVID-19 virus. They were tested with this machine and they are more successful with this 3D printed. They have checked and the 3D printed was more close to reality.
Sir, what is the cost of this machine?
Shivani will tell in better way. See, we will not provide the exact cost. We will provide approx because what are the differences? If you are using just one example so that you get idea. If you are going to use thermoplastic printed, I think that thermoplastic printed itself costs something similar or something. If so, someone is using this machine, pneumatic printer. If someone is interested in thermoplastic printed, like that, will ask for the cartilage-like materials. So that will be direct of like cost will be super. Like you have to have that so we cannot. There is one basic machine cost. I think Shivani will tell you better way. I am not the approximately BioX6 how much BioX this one 50 to 50 like 5 lakhs one standard setup you can get this and for 90 lakh I think BioX6.
So with raw materials?
Raw materials are not too costly. They are already optimized. The only thing I think it should be available from selling advanced raw material. The raw material is not too costly. The machine cost, the hardware, software, because they are sophisticated, that's what they cost. Any more questions, please? I hope I was able to pass some message on 3D by 20. बीच में कुछ लोग सो गए थे मुझे ऐसा लगा सब ओके ऐसा होता है बहुत इंटरेस्टिंग है थोड़ा सा टेम्पो मोमेंटम हो जाता है सिग्नल डाइवर्ट हो जाता है मैं भी मोनोटोनस हो जाता हूँ बीच में थोड़ा सा एनर्जी आ गई थी जब इंटरेक्शन स्टार्ट करा I think if we have no more questions, sir, okay, thank you.
Thank you so much, sir, for this informative session. Sir have very well explained what is 3D bioprinting, what is its application, why do we need it, and how it works. I hope this is beneficial for all of us and we can use this in our field area, research area. So thank you so much, sir. Now it's lunchtime. Now you all can proceed for lunch in the ground. And after lunch, we will meet again at 2 p.m. sharp for the hands-on training. Thank you.
You all are requested to please take your lunch in a very systematic manner. Please do not rush.
Sir, because if we start